Welcome to this special podcast from the Triple Helix Cambridge Café Scientifique, sponsored by the Medical Research Council. I'm Mira Senthilingam from thenakedscientists.com. The January Café Scientifique was about pandemics and the science of emerging infections. It aimed to give an insight into what emerging infections are, which ones we should worry about, and how we can go about tackling them. The speaker was Dr. Chris Smith, virologist at Addenbrooke's Hospital.
As you've heard, I'm Chris Smith. I'm a clinical lecturer in virology, and my specialty is in clinical virology, so diagnosing people with horrible things. And one of the horrible things we're diagnosing people with are flus. Very, very common every winter, and we've obviously come through the first pandemic that many of us will have seen in our lifetimes. Not everybody, but most people in our lifetimes. This is the first time we've actually seen the entry of a new pandemic. But let me ask you a question, because as soon as you say pandemic, everyone thinks flu. But how many people in this room actually would like to volunteer a number for the number of people who die every day of HIV?
10,000.
What do you think of 10,000 as a number?
Too high.
You're saying lower. Shout some more numbers. 5,000? You're not far off. 7,000 people every day. Okay, so every minute, in the roughly a couple of minutes I've been speaking, that means 10 people have already died of HIV. And it's not yesterday's problem, because every minute 5 more people get infected. And for those 5 people, that's actually a death sentence, because the majority of them are in sub-Saharan Africa, where they have no prospect of getting any kind of treatment. So when you say pandemic and everyone thinks flu, you think, oh, flu nasty. The reality is that flu is fairly trivial in the grand scheme of things. It's a nasty virus, and perhaps 500,000 people a year die of flu all around the world every year. But it's trivial compared with HIV. HIV is the worst pandemic that we've ever seen as a species. And with those sorts of numbers, and millions of new infections every single year, it eclipsed flu a long time ago. In just 30 years of existence, flu's been around for thousands of years. And the really interesting thing is, if you wind time back a little bit to the late 60s, early 70s, there was this huge optimism around the world because everyone thought we have solved the health problem. William Stewart, who was the US Surgeon General, a man with a big wallet, he had a lot of money to spend because he could decide where budgets got applied. He actually went on the record and said, it's time to close the book on infectious diseases. We need to concentrate on chronic ailments like heart disease and cancer. The war against infectious diseases has been won. There was a guy from Australia who won the Nobel Prize called Macfarlane Burnet. He was a very eminent virologist, and he in the 1970s said, the future of infectious diseases is going to be terribly dull. He's very wrong. Because we are cruising for a bruising. It's a miracle, many people say, that we haven't actually already wiped ourselves out with something. And when we say pandemic is just an infection that involves the whole world. In other words, it starts somewhere and spreads to involve everybody. And HIV is a really good example of that. We know when HIV appeared. It appeared in about the year 1900. How do we know that? Because people have found old blood samples. They even found old tissue samples in Kinshasa, in the Democratic Republic of Congo, in a basement, embedded in wax. And by dissolving the wax away and getting the tissue out, they could get RNA, the genetic material, from the virus from it. And by looking at the genetics of that virus, you can wind the clock back and work out how old it must be and work out how it compares with modern-day HIV. And because you can compare the genetic differences, you can work out how fast HIV is evolving. That's how they know when HIV first appeared, about 1900. So it's an example of what we call an emerging infection. And that's what pandemics are. They're emerging infections. They're infections that have either appeared new on the scene from somewhere, or they already existed, but in a very confined, restricted part of the world's surface, and they've suddenly started to spread. So what makes that happen? Well, really, you can distill it down into 3 things. Something that the host does, in other words, us. Something that the pathogen does, in other words, the thing that's making us sick. Or something that the environment does. And those 3 things, either in isolation or in combination, in any combination, can explain most of these pandemics. So what do humans do? Well, how many of us are there on Earth?
6 billion.
About 6.7 billion people on Earth. What's the population increase annually? It's about 1%. Doesn't sound like much, does it? I bet you're not getting about 1% on your savings. Are you impressed with your savings rate? No. But the reality is, if you had your 1% in the bank for 70 years, you'd have doubled your money. So every 70 years, at a growth of just 1%, the Earth's population doubles. So there are more people alive now than there have ever been. And as a result, we are cramming more people into less world than we've ever done. More people are living closer together than they've ever done. And more people are more mobile than we've ever been. How many people are airborne around this planet right now? How many people are up there in aeroplanes, not counting the people on the International Space Station? A government working party report looking at climate change in the year 2000 calculated there were 500,000 people airborne around the Earth at any moment in time. Aircraft travel is rising at 10% per year. So we can conservatively say there's at least 600,000 people flying at any moment in time. If you look at that kind of dynamic, if I wanted to travel 100 years ago from where Kate at the back lived and come here, how long would that journey have taken me?
Depends where she lived.
Australia.
6 weeks? 100 years ago, 6 weeks, she'd have been lucky.
4 months.
It would have been
6 months.
months to a year. It would have been months to a year by boat. Very long trip, very long time. So if I caught something in Sydney and then started coming home, what would be the outcome by the time I reached London?
You were dead.
I was either dead or better. Now, I get on an aeroplane, I can get to any major centre, anywhere in the Earth, in less than 24 hours. Because no major city is more than 24 hours from any other. The incubation period of most major infectious diseases is more than that, including flu. So in other words, I can pick something up in one part of the world, I can fly to a totally different one, and I don't even know I've ferried a virus from there to there. So this level of travel, this level of population flux, is a major risk. And with population density increasing, this means the opportunity for things to spread is also enhanced. How else are we increasing our risk of giving people things? Coming to social gatherings. So people live in big populations now. Thousands of years ago, small clusters of people lived in small clusters and they never got together. Flu first appeared as an infectious entity in humans about 5,000 or 6,000 years ago. What happened about 5,000 or 6,000 years ago? Why did we start to build cities?
Agriculture.
Because we have farmers. Why did we get farmers? Because if you have farmers, you don't have to spend your entire bloody life trying to find food, do you? Someone makes it for you. So you can go to the shops and buy it. Meanwhile, you can devote your time to doing something else that you're better at. And that's where a city comes in, because farmers support cities and cities support industries. Without your farmers, you can't have a city. But once you've got a city, you've got disease, because you give things to each other. And also, farmers keep animals. What sorts of animals do they keep? They keep birds and chickens, wildfowl, things that we can eat. Where did flu come from? Flu is originally an infection of aquatic birds. So by farmers getting established and then keeping birds in large numbers and putting those birds close to humans, that facilitated the opportunity for a virus carried by those birds to jump into the people. And that's how flu got started. Until people began to build cities and farms, we didn't have problems with flu. How else do humans and human behaviour cause us to get sick? Did cavemen have intravenous drugs? No. But nowadays, a very high proportion of people in cities, compared with yesteryear, inject drugs. And they don't inject drugs using their own sterile equipment. They're desperate. So they'll go, I need a hit, can I borrow your needle and syringe? What's on that needle and syringe? A tiny virus, hepatitis C. Very rare originally. Along comes intravenous drug users, and now 1 in 3 of them to 1 in 2 of them have got hepatitis C, all over the world. So, in other words, human behaviour creates niches that infectious entities that can become pandemics can fit into. There's now 200 million people around the world who've got hepatitis C. We also give people blood transfusions. Anita Roddick, who died recently, who set up the Body Shop, went on the record as saying, I've got hepatitis C. Where did she get it from? Probably during a blood transfusion when she was giving birth to one of her children. Now, we didn't know hepatitis C existed. We were just giving people blood, one person to the next. We didn't test for it because we didn't know it was there. So we began to transmit these things. We created an opportunity for an infection to spread. What about sex? Can't live without it. And in order to have children, you've got to have unprotected sex. There's a niche there. There's a microbe to take advantage of it. So human behaviour and human numbers create these opportunities. Even the credit crisis has created opportunities for viruses. If I tell you that there's a graph which shows the house repossession rate in California a couple of years ago, and it goes up in a straight line, very, very steep graph. And there's also reports of the number of reports of West Nile virus, which is a mosquito-spread disease, goes up directly in parallel by 300%. The housing repossession rate, 267%. The mosquitoes and West Nile virus results, 267%. Why are the two so closely matched? Well, if I showed you an aerial photograph of bits of California where lots of household repossessions occurred, what would I see in the gardens of those houses? Swimming pools. And if you look at the swimming pools, the houses that have been repossessed, what colour are they? They're green. Because no one's putting chemicals in them. And if you stop putting the chemicals in, they go green. The green is algae, what a mosquito larvae eat. So even the credit crisis and house repossession can give an opportunity to a microbe, which then becomes an emerging infection and spreads around the world. West Nile virus didn't exist in America until about 2000, 1999, 2000, it was first seen there. How did it get there? Because someone decided to move an animal, probably from Egypt. It was a bird that got shipped to the Bronx Zoo, and that bird brought with it West Nile virus. There were lots of mosquitoes around that year because it had been a warm spring, lots of rainfall, and the mosquitoes ferried the virus from that bird. First of all, it's in New York State. All the crows died. People thought, wow, what's this? Oh, it's West Nile virus, something we've known about for 50, 60 years. Used to be in a little patch of Africa, and now it's across the whole of the USA. At peak, 10,000 declarations of cases and hundreds, if not 1,000 deaths in 1 year from that. So, in other words, human behaviour is very, very important. Now, what about how we change bugs? How do bugs change? Well, the answer is they mutate with viruses. What are viruses? They're nothing more than an infectious bag of genes. And if you look at what viruses use as their genetic material, it's RNA. What's the difference between RNA and DNA?
Single strand versus double strand.
Absolutely. So DNA has two bits of genetic information. One is the mirror image of the other, and RNA is a single strand. So if you only have one strand of information, what can't you do? Yeah, you've got no RAID, have you? You've got no RAID array as a RAID 1, isn't it? If you've got one backup of one hard disk mirroring another. In other words, you have no backup. So if you bugger up your genetic sequence, you've no way of putting it right because you don't know you've gone wrong. HIV uses RNA as its genetic material. So how often does it make a mistake? The genetic sequence of HIV is about 10,000 genetic letters long. And the virus enzyme that copies it makes a mistake once in every 10,000 genetic letters. So every HIV particle, of which the average infected person probably makes 10 to the 11 per day, contains a mistake. So you can very quickly see why an infected person is making lots of different types of virus that they can start to spread around people. So the opportunities to disclose new forms of these things that can go all over the world and kill people, very, very high. And then you've got the environment. How might the environment impact on infectious disease, emerging infections and pandemics? What was everyone worried about in Copenhagen? Climate change. And why might climate change, apart from mosquitoes and things, why might that make a difference to the risk of infectious disease? Well, if you look at where the bugs and things are, if you change the environment, the things that carry those bugs won't like the environment anymore, so they might move. And if the bug goes, the parasites and the things it carries, the nasties, go with it. And the same applies to people. We think that if climate change goes to its logical conclusion, the amount of available land area on Earth could decline by something like 50%. Farmed, rainfall fertilised, livable land could decline by up to 50%. That means the population density is going to increase enormously, but not just for us, for other animals. Animals that can't tolerate the environment they may find themselves in, so they will move, and they will come into closer contact with people. And as a result of those contacts, they will pass what they carry onto us, or at least there's a better chance that they might. So let me just highlight those three mechanisms with a couple of real examples, because I've talked very generally. So let me give you a few specific examples so you actually believe that this is real. Now, who remembers what was happening in the year 2003? SARS. SARS was a nasty virus. It stood for Sudden Acute Respiratory Syndrome, or Severe Acute Respiratory Syndrome. Both names were used. Never been seen before. Popped up on the scene in 2003. Where did it come from? Geographically, first of all, it came from China. In fact, it emerged in 2002 in China. And it didn't actually emerge on the world stage until 2003. And what happened is that in Guangdong province in China, it probably first popped up. And then a person took it to Hong Kong, and that person actually was a doctor, ironically, and then jumped on a plane, and SARS got to 8,000 people. That's who got infected in 30 countries, and there were 800 deaths. What else was happening in 2003, out of interest? Anyone remember? Why did we invade Iraq? The idea was Saddam Hussein had weapons of mass destruction, right? Yeah? Biological weapons. Now, I was a casualty officer at the time in London, and we were doing weekly training where we'd have to blow up some tunnel, pretend that someone had died of VX poisoning or something in the car park, and ferry this person with anthrax or whatever in through this blow-up tunnel wearing spacesuits and chuck them in an isolation cubicle, and then demonstrate we knew what we were doing. People all over the world were doing those kind of things. When should we have been better able to contain an outbreak of an infectious disease than in 2003 when we were about to go to war with Iraq? Did we stop SARS? No. So there's the evidence that we really are potentially in trouble. Where did SARS come from? Actually, people thought civet cats. Now, the civet cats are actually quite big. And when the initial epidemiologists looked, they said, right, the people who've died of SARS so far, what have they been doing? Let's look for common factors. And civet cats cropped up. So then people thought, well, let's kill millions of them, and that will stop the outbreak. If civet cats have brought SARS to humans, we'll get rid of them, and that should stop it. So they culled huge numbers of civet cats. Did that work? No, it didn't work. But by then they already had some isolates of SARS. So they then infected the civet cats with SARS, and what do you think happened to them? Well, the answer is they died. Is that a good sign of a carrier? No, because carriers carry something without looking overtly sick. So that told them it can't be the civet cat. So the WHO, a year later, went hunting around. They went and did an expedition into rural China. And they actually went to some caves, and they just tracked everything that moved. And they found some bats, rhinolophus bats, horseshoe bats. And they found the immediate relative of SARS living in those bats. So now there's a conundrum, isn't there? You've got a virus that lives in a bat, and somehow it got from a cave in the middle of nowhere into Hong Kong. How did that happen? Well, no one knows for sure, but what they do know is that those bats routinely crop up in markets in Guangdong, in that province. And so do civet cats. So the argument is probably husbandry techniques, the way we keep animals. Someone put a cage full of bats and a cage full of civet cats side by side in a market, and one shared its infection with the other. And the civet cat amplified the infection, and being a mammal more closely allied to a human than a bat, the infection was more humanised, the civet cat was more infectious, and it spread into people. Which just goes to show, if you have large numbers of people moving large numbers of animals and putting them into configurations and contacts that they shouldn't be in, you give these viruses an opportunity.
That was Dr. Chris Smith, virologist at Addenbrooke's Hospital, explaining how the war against emerging infections may never be over, but with careful monitoring, education and continued research and development of drugs, we may be able to manage them better in the future. After the talk, there was a chance for audience members to get their questions on the topic answered.
So where did SARS go?
SARS didn't completely disappear, because it persisted in those bats, but also it did come back a bit the next year, but only in a very limited way. The thing with SARS is that it wasn't actually that infectious. So as soon as people realised what it was, and it was actually the first time anyone had ever seen a virus which, within just a few weeks, we had the complete genetic sequence for, that meant they could have tests that were specific for it. We understood the infectivity, and cases could be tracked and isolated, and contacts of those people isolated, and that meant it was much easier to break the back of it and control it. So once the epidemic, the pandemic as it became, was controlled, it was then easy to stop it going any further. And it was that initial outbreak that caused the problem. It did disappear partly because it was controlled, and also probably because it was less infectious than, say, flu. Doesn't mean people aren't still looking for it, and in our lab, for example, as part of our respiratory panel of tests, when someone comes in with an upper respiratory infection, they get tested for SARS. They don't know that, but they get tested for SARS. We're just screening for it because these things have a habit of just drifting around in the background and then suddenly surging. The other point is that lots of people became immune, and once you've got lots of immune people, your so-called herd immunity is very high, and that means that the number of people in the population who are susceptible is low, and if there are very few people who are susceptible, the chances of an infection getting onto somebody and successfully infecting them and them passing it on becomes lower.
With the swine flu, I mean, it was the big scare and then it wasn't the big scare. Should people have been as concerned, and who created the big scare in the first place?
There's one school of thought that suggests that swine flu was man-made. Is anyone shocked by that? When people sequenced the virus genetically, and they began to say, well, when did the flu that made swine flu last circulate? Because swine flu is a cocktail of flu viruses. There's a bit of bird flu, a bit of swine flu, and a bit of human flu in it. In fact, to make it, 3 different pig viruses, one from Asia, one from Europe, and one from America, had to come together and combine. And the only way that would have happened is if someone took a virus from all those 3 different locations and mixed them together in a pig. Either quarantine broke down 3 times over at all the same time and enabled this to happen, or there's another explanation. The other explanation is that samples of flu are routinely shared by pharma companies all over the world all the time because they're used to make vaccines. They're used to make vaccines to protect animals. So cells and virus itself are being sent all over the world. And one suggestion is that one day someone was making a batch of flu vaccine for a pig, and they forgot to dump the inactivating chemical into this vat of flu virus. So when they inoculated a pig, they actually injected it with a whole bunch of live viruses. As a result, the pig was productively infected, and this acted as a mixing pot, and as a result, it disclosed a new form of flu that then spread from the pig into people. The evidence for that is that when people look in the database and say, well, when did those strains of the flu that are in swine flu last circulate all together around the world, at least 10 years, and in some cases 17 years ago is the answer. So why should something that hasn't been seen for 17 years suddenly appear alongside something else that hasn't been seen for 10 years, all in one place, and then suddenly start to spread? So many people are very concerned that this could be a farmer-driven outbreak, and they want to see better regulation in future. Whether or not it was a real worry is that it's easy to be wise in retrospect. What we'd seen from bird flu, H5N1, is that that's nasty. About 70% of people who catch it, compared with, say, 0.5% of normal flus, those people were dying. So the government were worried that if a pandemic flu appeared here, we might see 70% mortality rates. So all of the plans that were made, the prophylactic drugs that were put on hold, on ice, Tamiflu, those were all geared up with H5N1 in mind, severe bird flu type flus. What came to town was actually a much softer thing, and it's a bit like going to war against natives armed with spears, and all we had were nukes. And it's a question of, well, what are we going to do? We haven't got anything apart from nukes, so we'll have to nuke them. So they did. Whether it was the right thing to do or not, we don't know. Some people are now concerned because what we see in the UK is the highest rates of Tamiflu resistance of almost any country worldwide. And the suggestion as to why this has occurred is that when the flu first appeared here, the first thing we did was to blitz it with Tamiflu. What that probably did is to select out from the flus that were spreading around the strains that were best able to spread in the face of Tamiflu. Because, like I said, viruses make genetic mistakes. Flu is no exception to that rule. And as a result, we ended up with flus spreading around that were Tamiflu resistant to a certain extent, or had the ability to become resistant. And now we're seeing the results of that.
What would be your top 3 recommendations for better protection of humanity from the terrifying scenario that you were describing in your presentation, where we're all doomed?
I think at the bottom line here is population. And the best protection we can buy for ourselves is to stop the population of the world getting any bigger. Because whenever there's a war, because people are fighting over available resources, there are outbreaks of infectious diseases. With climate change, which is directly linked to how many of us there are, because every single one of us puts out between 4 and 6 tons of CO2 every year, and that's just to live in a Western country, and the people in less developed countries are increasingly beginning to follow the same lifestyle that we have had, which means that the potential contributions of CO2 is going to be huge. That will lead to inevitable conflict because people are worried about land area, they're going to worry about where their food is coming from, and as a result this will have all these other problems. So I think probably getting to grips with population should be our number one priority, because that's the one thing that's going to be the biggest driver of infectious disease.
Please could you say something about the 1918 influenza pandemic?
Well, 1918 came at a very bad time for the world, because it was straight off the back of World War I. And this probably helped it get started. We actually think it started in America, But the one thing that probably facilitated it, regardless of where it started, was that there were thousands of people, all billeted around the world, all living in bad conditions, on top of each other, in barracks, and they were very mobile. They started being shipped back to their home countries. And they were also poorly nourished, and so were the countries that those people went back to, because many of them had been ravaged by war. And as a result, people were less able and less fit to defend themselves immunologically. And this virus that appeared that no one had seen before gained a toehold. And the reason it was so successful is because this Spanish flu, H1N1, had never been in the human population, or at least not in the lifetime of many of the people alive. That meant that none of them had any kind of immune defence. They had no antibody against it. So as soon as you saw it, you got infected. Very high infectivity rates. And that meant that everybody who got infected gave it immediately to very large numbers of other people because they were all susceptible. So the whole thing mushroomed. And as a result, lots of people died, even though the actual mortality of the virus was quite low. It was less than 2%. But the people who were dying tended to be younger people. There are probably two reasons for that. Older people may have seen H1N1 before, and they may have had some underlying immune defence. In other words, they had antibody that sort of recognised H1N1 a bit, and that protected them a bit, enough to mean they didn't die. But young people fall into two groups. Very, very young people, children, who are more vulnerable anyway, and then individuals who are fit and healthy, hopefully like me. The problem is that people in that category have a very good immune system. And what H1N1 seems to have been doing under those circumstances is driving a very, very florid immune response. So it got deep down into the lungs, and it then drove the immune system nuts. And what happened is the immune system was attacking healthy tissue in the course of trying to eliminate the virus. And in the course of doing that, damaged healthy tissue, causing people's lungs to fail, and they developed respiratory failure. And that then caused people to turn blue and die in 24 hours in some cases. And it was principally young people with healthiest immune systems that happened to. These hypotheses are being tested because scientists have been able to rebuild that virus, because tissue samples kept on microscope slides exist in London and a few other places, and whole bodies of people who died of that flu have been locked in the ice in Alaska for almost 100 years. And those bodies have been thawed out, and samples of the virus is extracted. And by using the polymerase chain reaction, PCR, it's possible to rebuild the virus, because you can work out what the genetic sequences are and link them all back together again. And in the last 5 or 6 years, that's exactly what's been done. And in a containment level 4 laboratory in America, this virus has been introduced to mice to see what happens. And because flu has got lots of different bits that make the virus, researchers are taking different combinations of those genetic components and trying them in different combinations to work out what the particular lethal cocktail of H1N1 was. But we think it's because it drove the immune system totally wild in people with a healthy immune system that it was so lethal.
Would it have made any difference if then we'd had antibiotics?
It's quite a good question because, of course, flu is a virus. So the knee-jerk reaction to are antibiotics any good against a virus would, of course, be, well, of course not, because antibiotics kill bacteria. But flu's interesting because it kills people in two ways. Some people get an acute influenza pneumonia, and the virus actually makes its way down into the lungs and attacks lung tissue and kills people. And under those circumstances, probably antibiotics would make virtually no difference. But a second group of people develop bacterial superinfections. The flu injures the airways, and this opens up gaps, sort of immunological niches, that bacteria, which naturally are carried on us, can access and then exploit. And actually the most common cause of this happening is Staphylococcus aureus, the thing that causes impetigo, and about 15% of people have it living in the front of their nose, in their armpits and in the crotch. Anyway, so I'm not saying shove your nose in someone's crotch to guarantee you've got it, but the vast majority of people have Staph aureus, or a large number of people come into contact with Staph aureus, and all you have to do is sniff quite hard, and you deposit some of these bugs down into your lungs, and as a result, if you've got injury there, you could get them. And so those people who died of staph pneumonias, for example, could have been rescued, potentially, But usually people who get into that position are so ill anyway, the mortality rate from that's already really high. But it probably would have made a bit of difference.
When you say, in one of your examples, you talked about all the crows dying. I'm wondering what proportion of the crows really died, and how, in human infections, the sort of resistance to, or the people not dying is a result of previous exposure to similar viruses, or how much of it is just purely down to their genomes not being compatible with the virus's way of acting.
Yeah, this comes down to the question of, well, what's a carrier? Well, a carrier is someone who genetically is well adapted to carry something and not get sick. Because the ultimate and ideal outcome for a virus infection is that it will make you ill enough to be infectious, but not so ill that you don't go about your business and infect loads of other people. Because what you want if you're a virus is you want a person to be really infectious for as long as possible, but go to work, go and party, go to the disco, snog loads of people, give yourself to them. Now, that's not going to happen if you're moribund and dead too quickly. So you've either got to be really, really excruciatingly infectious and make someone so infectious that they give it to everyone a mile away, or you're not too virulent so they can still pull themselves out of bed and struggle into the office, but you're pretty infectious, so once they get there, everyone else in the office is buggered. That's the ideal outcome. Now, over time, viruses will evolve to become more like that, because if you think of it in terms of numbers, if you start off as really, really fatal as an infection, you will kill loads of people. And the people that are left will be either immune, or they're not going to get infected by you, or there's going to be so few of them that you've got no chance of spreading. And measles is a really good example. Measles has only existed for about 5,000 years. It came originally as a disease of cattle called rinderpest. That's the closest relative of measles. So what's this magic 5,000 number? Well, it's not so much farmers, although they would have been keeping cows and probably that facilitated it, but what measles needed was big populations. You need, with a smash-and-grab type virus like measles, you need a big population to sustain it. We think something in the region of 10 to the 8 people, 10 to the 6 to 10 to the 8 as a global population to sustain something like measles. That sort of population level didn't exist until relatively recently. So those viruses just didn't gain a toehold. To take a more contemporary example, if you look at HIV, there are people who emerge in the population now who are resistant. Have you heard of these individuals? And you can challenge them with HIV and they don't get infected. And this comes down to a particle or a structure on the surface of white blood cells. Because HIV, to get into white blood cells, uses something called CD4. This is a protein on the surface of a certain class of white blood cells. And the virus has got something called GP120, which it uses to lock on to CD4. But that's not the whole story, because the virus also needs to grab hold of cells with its other hand in order to help it get in. And the other hand that it grabs hold of them with is it uses this thing called CCR5, which is another chemical on the surface of a white blood cell. If you look at these people who you cannot infect with HIV, they've got a bit missing from their CCR5 protein. And these people have a gene called CCR5 delta 32. And these individuals, you can challenge them with HIV and they're very resistant to infection. The virus just doesn't seem to be able to get into their cells. And it turns out that a fairly high proportion of the population of Europe, not Africa, but Europe, has this gene. So why should a deletion in a gene be so common in Europe? Well, if you look back in history, we had the Black Death. And a third of the population of Europe died. And if you look, anecdotally, in places where the Black Death was most manifest, in other words, where the death rate was really high, the populations still surviving in those areas are more likely than expected to be by chance carrying CCR5 delta 32. And what this suggests is that the Black Death in some way also picked out people that didn't have that gene. And it selected out people who did. And the gene that protected people against the Black Death 500 years ago is now protecting people against HIV today. So that explains how some people get genetically selected by a virus so that it can either coexist with them and not make them very sick, or they become genetically resistant to its effects. And if HIV was allowed to go to its logical conclusion without people chucking drugs at it, what would happen is that all the people that were susceptible to HIV in the short term would die, and we would then end up with a genetic stock of people who were more resistant to its effects or just became chronic carriers without really becoming very sick.
Could that inability to latch onto a protein be developed into a treatment for, well, or not a treatment, but a preventative for HIV?
Yes, and it has. People know what the natural ligand of that particular receptor is. In other words, what the molecule in the body that normally locks onto CCR5, they know what the ligand is. It's a chemical called rantes, R-A-N-T-E-S. So if you flood the body with that, when HIV tries to go onto the same receptor, the rantes is in the way. So people have been trying to use this as a way of blocking HIV entry at mucosal surfaces. So most HIV is spread sexually. So if you cover the mucosa of the genitals with ligands that look like that, then you can potentially compete with the HIV and stop it going in. And that's exactly what people have been trying to do. And that was the first way of doing it. Now, there are 2 new agents that have just been released. One of them is called Miraviroc, and another one is called Vicriviroc. And these are actually drug molecules that have been rationally designed to selectively block those 2 receptors. And they're being used in patients right now as antiretroviral agents. So you're spot on the money.
You were saying before that reducing population would be a good way to prevent viruses, but if we assume that's not likely to happen, do you think there's much chance that advances in medical technology will save us all?
You could say, if we had infinite amounts of money, yeah. But the reality is that the vast majority of the world don't have access to those kind of resources and don't have access to the right money. Therefore, they're not going to be saved. So I don't honestly think that, apart from in an ideal world, that medicine's going to be the answer to everything. Because we just can't afford it. Not in the short term, anyway.
How does a drug like Tamiflu work? Because somehow we got this idea that antivirals are quite difficult because we know there's not much you can take for the common cold, but we understand antibiotics. So I don't understand how a drug like that works.
Oh, it's really simple. What happens is that Roche make this agent, and then they put a big price tag on it, and then they tell everyone that it's the only thing you can use, so you've got to buy it, and then their share price goes up. But as they own all the shares, because it's a private company, then they all rub their hands together and then tell you Tamiflu is great, so you buy a load more. That's how it works. What it actually does pharmacologically is it's not actually clinically that effective. It's certainly not the viral equivalent of penicillin tackling a bacterial infection. The way it works is it's what's called a neuraminidase inhibitor. And if you look at a flu particle, they're very tiny. Flu's about 100 nanometers across, so that's about 1/10,000th of a millimeter. And on the surface of this particle, which looks like a spiky meatball, you see these 2 things sticking off. You've got mushroom-shaped things and kind of spiky things. And the spiky things are hemagglutinins, and they lock on like viral Velcro onto your cells. And the mushroomy things are an enzyme, neuraminidase, and that chews holes in snot. The other thing it does is it chews holes in sialic acid, which is what the hemagglutinins is locking onto. And it's the way the virus gets out of an infected cell. So when the virus is made in a cell, it buds out through the membrane of the cell, but to get free, it chops all these anchors to the cell surface, and that's what this neuraminidase does. If you block up the neuraminidase with a drug like Tamiflu, it impedes the ability of the virus to leave a cell and therefore infect other cells and other people. The problem is that viruses mutate, and what we're seeing is that the virus has changed the shape of its neuraminidase a bit. There's one mutation it can make, which is becoming a bit more common, which changes the structure of the protein a little bit so the drug no longer effectively disables it.
Are there many antiviral drugs out there? Because Tamiflu existed for a while, didn't it? So when swine flu came, we'd got something to call on. Do we have a nice bank of things to call on, or should we be worried?
How many people have got a cold in this room at the moment? Probably everyone should have their hands up, because the likelihood is we've probably all got something all the time. So viruses are really good at spreading. They're really good at making you sick. They're really good at not making you sick, just spreading. When people have done tests, they've asked people, have you had flu this winter? And all the people that said no, they then took blood samples from them and asked the same question immunologically. Are there antibodies in this person reflecting recent infection with flu? And the amazing thing was 50% of those people who said no had antibody responses that indicated they have had the flu recently. So that shows that lots of people get flu infections and don't even know they've had it. So these viruses are really common. And the fact that they're so good at spreading makes tackling them and targeting them very, very difficult. Viruses are molecular parasites. They're nothing more than a bag of genes that's very good at locking onto a cell, invading it, putting a genetic message into that cell, hijacking the cell and making the cell make thousands of new virus particles and then die. And as a result, it's not easy to find a molecule that will discriminate between the virus and the human. Because with bacteria, they're their own entity, they're their own cell. They use all their own machinery to make copies of themselves and to grow and to spread. And therefore it's very easy to discriminate against them because you can find differences between the bacterial cell and the human cell. Because viruses are, by definition, using your own tissue, it's really hard to find a way to shove a spanner in the works without buggering up every other cell in the body. So that's why there is not this big pipeline of antiviral drugs. Well, the difference is with HIV, because there was such a huge problem there and such a huge spend went into it that there's a very big repertoire of drugs which can now target HIV, and they do it in lots of different ways because we understand the life cycle of that virus very well, and because the life cycle has various effectively critical components to it, there are various things which are absolutely essential that the virus has to do to spread. You can make molecules that will block up those particular processes, and they do it very well. And if you give people lots and lots of, or at least triple therapy, so 3 drugs all at once to block the virus from 3 angles, you minimise the risk of it mutating, and you therefore suppress it down to almost undetectable levels. And this means that the death rate with HIV, which was about 10 years out from infection, you could guarantee you would have an AIDS-like illness probably, that's been extended to 20, 30 years now. So HIV is being viewed as a chronic illness in people who have access to drugs. But for the majority of infections, like the ones circulating in this room, there is nothing you can do about it apart from Lemsip.
Well, again, less of a question than just to get your thoughts on current situation of tuberculosis. Would you call that a global pandemic at this point in time?
When I was thinking what I was going to say when I was standing at the back drinking orange juice, and I thought, well, TB, the numbers are not grossly different than HIV. The number of people dying every day is up to 10,000 from TB, and globally 1 person in every 3 is infected. If you go down to London on the Underground, the likelihood is you're going to be challenged by TB, if not infected, in 1 tube journey. So it's a major problem, and the majority of the disease burden is actually in sub-Saharan Africa, because most people who get TB will wall it off, and it will remain in their body but not active for the rest of their life. It may then recruit deaths and cause a problem in elderly people or in people who have immunosuppression because they have to take drugs to suppress the immune system. But another major cause of immunosuppression is this other massive pandemic we've harped on about all evening, HIV. And the reason Africa is such a problem is because these people catch HIV and then all these other mycobacteria, not just TB, but other mycobacteria, members of the same family, then become a major problem for those people. So it is a major threat. It's a major priority area. It's been defined as a priority area by the Bill and Melinda Gates Foundation. It's one of these latent diseases, and people are very, very worried about it because it's getting worse. The numbers of cases in the UK is about 8,000 cases per year, which is huge, considering that it was down to half that and is now coming back up again. Part of that's immigration, part of it's alcoholism, homelessness and HIV, but it's certainly one to watch and one we should worry about.
So the audience were able to get into a good discussion with Chris on the night, which is the main aim of Café Scientifique. And to find out just how much the audience had gotten out of the evening, I spoke to some of them after the event to find out what they'd expected and what they'd learnt about emerging infections.
I was quite open-minded, really. I was interested to see how the subject might be tackled, and Dave certainly brought up some interesting points that, I mean, in a previous life I was a biologist at university, so some of the things I was familiar with, and when I sort of thought about them, it made sense.
And what would you say you've taken away then from tonight's event and learnt about viruses and pandemics?
Well, really how I wasn't aware that the economic situation the world's facing at the moment could contribute to pandemics. It was very interesting hearing about how neglected swimming pools from repossessed houses were becoming a reservoir for algae that was then having a knock-on effect for invertebrates spreading diseases in particular.
So things that you just really wouldn't have thought of otherwise?
Exactly, yes.
I've been to some similar events, like part of the Science Festival and things, and I'm not a scientist. I just like to come along and find out what's going on and, you know, hear a different perspective on the world. So.
And what did you pick up then today? So what did you think of today's perspective?
It was really accessible. Some of the very scientific side of things I didn't quite grasp, but enjoyable, accessible and good speaker.
Has it left you concerned about emerging infections?
Yeah, we can't worry too much about that.
So I knew it was going to be about pandemics, and I thought, well, swine flu's been the most recent, so probably be quite a lot of focus on that, but it was actually broader than that. It was a lot of other infections and viruses as well.
What did you learn about them, and are you now worried about infections?
It was brilliant. I really liked the amount of detail that we could get into because Chris knew so much, and so I got out of it a lot of other questions I couldn't get answered just by watching news reports. So they were quite detailed, and I don't understand the detail always, but there were things like, how does Tamiflu work? And it was quite a detailed answer, but I was happy that somebody could actually talk me through the mechanism, and I really enjoyed that level of interaction.
Was there anything surprising that you learnt today about infections?
I think it was really good that Chris kind of awoke us to issues like HIV and the number of people that's still killing every year and the number of infections that still are every day, because I think in a country like this you can still be very fixated on quite trivial things. I mean, there obviously people die from swine flu and things like that, but it feels a bit like we're quite worried about various sort of respiratory diseases, and actually we've kind of lost track of, in Africa, there are some other more like challenging conditions, still prevalent.
I was expecting to be engaged, which I was, to hear new material, which I did, an interesting mixed audience who would ask interesting questions, which they did, and we would get knowledgeable answers from somebody who knows what they're talking about, which we did.
So all your needs were met?
Absolutely.
And would you say you learnt anything particularly interesting or surprising about pandemics or emerging infections this evening?
Well, I'm a teacher of A-level, so what I learnt was some examples to use, some statistics which I can use, so I can slip those into the teaching. So it actually helps the students as well as me.
So the audience seemed to appreciate the level of detail Chris had gone into, and as a result have a better awareness of infections, varying from the common cold and the flu to HIV and tuberculosis. So you could say the science itself was infectious. Now to wrap up, I caught up with Jenny Malloy, co-organiser of the Cambridge Café Scientifique, to find out what we have to look forward to at next month's event.
Well, next month we've got Dr. Carolyn Crawford, who is Outreach Officer at the Institute of Astronomy, coming along to give a talk entitled Our Place in the Cosmos, where we'll be exploring all sorts of wonders of the universe, and that's at 7:30pm on the Wednesday the 24th of February in the Larkham studio at the ADC.
And you can find out more about that event online at cafescientifique.org/Cambridge. So come along in February to the ADC Theatre in Cambridge and join in to find out more about our place in the cosmos. The Triple Helix Cambridge Café Scientifique is sponsored by the Medical Research Council, and this podcast was produced by me, Meera Senthilingam, from thenakedscientist.com.